Good day, this is Professor Will, CBMD PhD, and today is Thursday, July 22nd, 2021. It is 2:25 and 8 seconds, p.m. Eastern Standard Time. Today's research, heart model. So let's hop to it here. Dynamic heart model mimics hemodynamic loads. Advances engineered heart tissue technology. Date July 21st, 2021. Source College of Engineering, Carnegie Mellon University. A new model mimics physiologic loads on engineering heart muscle tissues, yielding an unprecedented view of how genetics and mechanical forces contribute to heart muscle function. In efforts to understand cardiac disease progression and develop therapeutic tissues that can repair the human heart are just a few areas of focus for researchers. The research laboratory has been working for a long time on engineering and building human heart muscle tissue so they can better track how disease manifests and also create therapeutic tissues to one day repair and replace heart damage. One of the challenges is that they have to build these small pieces of heart muscle in a petri dish, and they have been doing that for many years. What they realized is that these in vitro systems do not accurately recreate the mechanical loading they see in the real heart due to blood pressure. Hemodynamic loads are the preload stretch on heart muscle during chamber filling, and afterload when the heart muscle contracts, are important not only for healthy heart muscle function, but can also contribute to cardiac disease progression. Preload and afterload can lead to maladaptive changes in heart muscle, as is the case of hypertension, myocardial infarction, and cardiomyopathies. Researchers introduced a system comprised of engineered heart muscle tissue, EHT, that is attached to an electric strip designed to mimic physiologic preloads and afterloads. The first of its kind model shows that recreating exercise, like loading, drives formation of more functional heart muscle that is better organized and generates more force each time it contracts. However, using cells from patients with certain types of heart disease, these same exercise-like loads can result in heart muscle dysfunction. Researchers treat patients that have genetically linked cardiovascular disease, including a type called arrhythmogenic cardiomyopathy, ACM, that often becomes worse with exercise. They have been able to get patient-specific induced pluripotent stem cells, differentiate these into heart muscle cells, and then use these in their new EHT model to recreate ACM in a petri dish so they can better understand it. Looking to the future, the collaborators aim to use their model and findings to study a wide range of other heart diseases with genetic mutations, develop new therapeutic treatments, and test drugs to gauge their effectiveness. Researchers can take lessons learned from building the EHT in a dish to create larger pieces of heart muscle that could be used therapeutically. By combining these new results with their previous work involving 3D bioprinting heart muscle, they hope to one day engineer tissues large and functional enough to implant and repair the human heart. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.